Tamso Ma Jyotirgamya, leading from darkness to the light of knowledge. Good morning, everyone. Myself, Dr. Sakshi Bhardwaj, on behalf of Elton Technologies, it's my pleasure to welcome you all in this subsequent session of Women's Day special webinar series on Molecule to Medicine Women Leading Collaborative Science and Informatics. I'm thankful to all of you for sparing your valuable time to join us today. Moving towards today's webinar actually, which is on application of machine learning in miRNA based cancer diagnosis. You know, AI ML pertains to multiple sectors. Among them, drug discovery benefits greatly from AI ML, that is artificial intelligence and machine learning, which speed up the process of finding possible drug medicines or the therapeutics, predicting their toxicity and efficacy, optimizing lead component, and analyzing massive data sets to find new drug targets or a therapeutic component. This ultimately leads to faster and effective, I would say, the drug development process with the improved success rates. Means every step of drug research or drug development process uses AI ML. To know more about how AI ML would be beneficial in the diagnosis of a disease condition, we have our expert with us, Dr. Zainab Siddiqui. Dr. Zainab, working as assistant professor, Center for Disease Mapping and Therapeutic Research, Era University, Lucknow. She has completed her graduation and master from Jamia University, Delhi, and PhD from Dr. APJ Abdul Kalam, Technical University, Lucknow. She has received numerous awards and grants. She is a lifetime member of Indian Cancer Society, Uttar Pradesh, and Indian Science Congress Association, even of European Society of Medicine, Medical Oncology, TKM. She is also on the editorial board of many respected journals and a member of scientific development committee for the investigating anti-cancer properties of natural compounds at the Asian Pacific Journal of Cancer Prevention. She has been invited a guest of honor for multiple times. Her credential also includes two book chapters and more than 14 publications. I welcome you, ma'am. I request all my viewers to feel free to drop their queries into the chat box. We will be taking question and answer after the session or end of the session. Yes, now without any further ado, I would like to hand over the platform to our expert. Over to you, ma'am.
Thank you, Dr. Sakshi, for inviting me once again to this Women's Day series that you host annually. And it's a privilege to be a part of such an inspiring event. This year's International Women's Day theme is hashtag Accelerate Action. So let's accelerate our research and integrate new technologies like machine learning into our data sets. So let me begin. Today, I will be discussing about applications of machine learning in miRNA-based cancer diagnosis. So this term machine learning was coined by Arthur Samuel in the year 1959. He worked on the player checkerboard, which improved itself by self-learning, and he is considered as the OG of machine learning. So machine learning is basically a branch of artificial intelligence, and the primary objectives of modern machine learning are prediction and pattern recognition. By prediction, we mean that if suppose I'm having a microRNA expression data, so through that data, I can predict if a patient is going to have cancer or not. Similarly, we can also identify pattern and relationship between data. So for this, we require large data sets that could be generated through different high throughput sequencing techniques. Then the steps which are involved in machine learning, let me give you a background check of these steps. First one is the problem definition. We clearly define the problem we want to solve. For example, we could focus on predicting whether a patient is having cancer or not based on the distinct microRNA expression profile. Next, we could define the problem to identify differential expression of microRNA at different stages of cancer. Another interesting problem could be predicting patient survival rates based on their miRNA expression data. Then comes the data collection, which involves retrieving relevant data from various sources such as databases and experimental data sets. As I earlier mentioned, it could be your high throughput screening methods like sequencing data could be your can be obtained from microRNA expression profile. Then this is followed by data pre-processing. It involves handling of missing values and data normalization. Next is the feature selection, which helps in the identification or extraction of variables of features like specific miRNAs that are important for cancer diagnosis. Once this has been achieved, we can choose an appropriate machine learning method. This could be anything from decision trees to neural networks, depending upon the complexity of your data. Now the model has to be trained and tested into training and testing sets so as to evaluate its performance. Then there is model evaluation, which assesses the model's accuracy, precision, recall, or other metrics based on the test data. Second last step is the model interpretation and explainability. Through this step, we understand how our model is making its predictions. This is very crucial, especially in sensitive fields like healthcare. Lastly, we have the model deployment step. Once validated, we deploy the model to production, allowing it to make real-time predictions or assist with decision making in a clinical setting. Now, coming to the data collection part, data can be of different types, such as it could be a clinical data, which is related to patient demographics, history, diagnosis, lab test, and treatment records. Then there is genomic data, which provides information like mutations, gene expression profiles, miRNA expression profile, or your sequencing data. Most popularly used these days is the imaging data, where medical images like MRI and CT scans are used for predicting cancer. Histopathological data has also gained attention for predicting cancer these days. Then there is electronic health records and publicly available data sets like TCGA, geo data sets are also there available, freely accessible to use by the users who are doing work on machine learning or artificial intelligence. Now, as today's talk is about miRNA based cancer diagnosis, therefore, one needs to know about the specific databases where we can find miRNA disease association. This includes MIR2 disease, HMDD, and DBEMC 3.0, OncoMIR. These all focuses on the miRNA disease association, while TCGA and the miRNA Cancer Atlas provide data on miRNA expression profiles in cancer. Then comes the third step, which is the data pre-processing. So once you have retrieved your data from the databases that I mentioned earlier, then you have to integrate your data. Suppose if I want to retrieve data from more than one database, for example, I want to integrate data of MIR2 disease and DEMC. So integration is very important, and for integration your data set should be the same. You have to harmonize the miRNA identifiers like those present in miRNA-based as IDs, and you have to ensure there is consistency across the sources. Once you have integrated your data, then you need to clean your data. Data cleaning means that you have to identify if there are any missing values or not. So you have to remove them by calculating the measures of central tendency, that is mean, median, mode. You can also identify the outliers by statistical analysis or using visualization techniques, for example, box plots. Once you have done this, then we move forward to data normalization. So the techniques involved in it are minimum to maximum scaling, where data is rescaled to a range of 0 to 1. Then there is z-score normalization. Here we subtract the mean and divide by the standard deviation for each feature. Then lastly, if miRNA expression levels span a wide range, suppose the lowest range is like 0.0001 and the highest range is from 20 till 25, or you can say till 50, then this is a wide range of data you are having about microRNA expression. Then you have to apply log transformation so that you can reduce the skewness of the data. Though shortlisting of miRNAs by differential expression analysis tools are also available. So what these tools do is they identify key microRNA biomarkers, and this feature selection is quite crucial to reduce the dimensionality and focus on the most relevant miRNAs. So these tools include DE-SEQ2. It identifies miRNAs that are differentially expressed in cancer tissues. Then there is EDGE-R. It identifies differentially expressed miRNAs between cancer subtypes like stage 1 cancer, or if there are several variants to cancer, like if you are having oral cancer, there is squamous oral cancer, there is adenocarcinoma. So you can differentiate the expression of miRNAs between them. Then there is LIMA. It also identifies miRNA with statistically significant expression in cancer. Then there is miR-D. Here you can analyze the RNA sequencing data. You can discover novel miRNAs and quantify their expression levels. Now, what are the branches of machine learning? Once we have done all, we have identified a problem, what we are going to look into, whether we want to segregate our patients based on the miRNA expression profile into cancer and non-cancerous or into specific stages. And then we have retrieved our data from the databases and we have pre-processed this. Now we want to perform machine learning, so we have to select a model. So depending upon our problem, if it is a categorical problem, for example, you say that it has cancer or not, then you have to go through supervised learning. If you have a continuous target variable, then you can perform regression models. These include the linear regression and the logistic regression, neural networks. Since you are having a categorical target variable, then you can classify your data through support vector machines, discriminant analysis, and KNN networks. In case your target variable is not available, you don't know which microRNA to target, which group to target, then you can perform clustering or association tools to develop a model. So here is a tabular representation of the studies going on in machine learning model development. There are several studies, and recently this tool has been identified in cancer diagnosis. For instance, there was a study conducted by Kopi et al. In the year 2024. He utilized machine learning algorithms to identify significant variations in miRNA expression between cancer patients and healthy controls. He highlighted the potential of microRNA 29A and 92A as non-invasive biomarkers for cancer detection. There are other algorithms also like random forest support vector machine, which have also been widely applied to predict cancer stages, as seen in the work of Urukela Hamidi. These scientists have worked in this area. There are also studies on how machine learning models can help in stage prediction, offering more accurate and timely diagnosis. Moreover, research conducted by Elizad and Hamidi, they emphasize the value of machine learning in assessing patient risk levels based on the miRNA expression patterns, paving the way for more personalized and effective treatment plans. So this highlights the current advancements in application of ML in cancer research and diagnosis, focusing on miRNA as an essential factor. So once you have developed a model, then you need to test your model and train it. For training and testing, you have to split the data set into the training data and testing data. Training data set usually includes 70 to 80% of your data, and the model will learn the relationship between the miRNA features and the cancer labels from this data. While the test data is of approximately 20 to 30%, it provides an unbiased estimate on how well the model can predict cancer in real world scenarios. Then comes the cross validation. So, cross validation, in this technique, you can perform cross validation by k-fold cross validation technique. So, by k-fold cross validation, you can divide your data into 5 subsets. So, 4 subsets will be doing the training, and only the one subset will go for testing, to ensure that the model performs well again and again when you are going through repeating the same process like 5 times. If you are taking k fold is equal to 5, then there is stratified sampling. If data is imbalanced, like there are more significant number of cases in non-cancerous sample than cancerous, the count is like you are having non-cancerous cases are like 100 and cancerous cases are like 50, so there is an imbalance in the data. So you have to stratify it, so there is a same proportion in both the groups of your data sets. Then once you have developed your model, you can evaluate it. So there are scientists not considering one model at a time. They can consider several models also. Like they can test their data set by on SVM model. They can test the same data set on KNN model. They can test it on the same data set on neural networks also, and identify which model is quite significantly assessing the problem that they are trying to solve in the first case. So the model evaluation, it involves key performance metrics. For example, first one is the accuracy. The proportion of correct prediction is important to identify the overall success rate. Then there is precision. The precision identifies how many of the predicted cancer cases are actually cancerous, so as to reduce the false positives. Then there is recall, how many actual cancer cases are correctly predicted to reduce the false negatives. Then there is F1 scoring system also. It creates a balance between precision and recall, and it is important for imbalanced data sets. As I mentioned earlier, if you're having more number of cases in non-cancer or cancer vice versa, then you can go for F1 score. Then there is AUC ROC curve, the area under the receiver operating characteristic curve, which gives a better understanding of the model's discrimination ability across various threshold. The higher the sensitivity and specificity value of the ROC curve is, the good your model is. Then comes the second last step, that is model interpretation and explainability. So in this section, what we do is we do the feature engineering, we do the hyperparameter focusing, then we do the model reselection. The first one is the feature re-engineering. So in this case, what we do, we identify the new relevant microRNAs, we shortlist them through feature selection and see how they are interacting with the target mRNAs. We can also identify their expression, like their upregulation, their downregulation in different cancer cases also, or within the same cancer in different stages of cancer. For example, a particular microRNA like microRNA 21, 145 or microRNA 146, these microRNAs were found to be dysregulated. As microRNA 21 is an oncogene, so what we can look into the pattern of its expression, like for example this microRNA 21 was slightly down regulated in stage 1 cancer, and as the cancer stage is progressing, this expression of microRNA is getting increased with the number of stages of cancer. So this pattern is identified through machine learning model, And similarly, it identifies similarly statistical difference between the patterns in all the microRNAs, and it predicts the accuracy of these interactions. Then comes the hyperparameter. We try additional values or combination to see if the model's performance can be improved. Then there is model reselection. For example, I tested my problem on a single model, that is random forest, and found that this model was able to predict the patient, the cancerous patient from the non-cancerous patient based on the microRNA expression profile. Now I want to see if there is another model which is far more better than the previous one. So I can try XGB boost model, and I can also combine both of these model through an ensemble and see how the combinational effect is on predicting the microRNA expression patterns in cancerous and non-cancerous. So once these steps have been followed and thoroughly done, then there comes the model deployment. You have designed your model, you have tested your model, you have trained it, and you have also checked its accuracy, if it's performing well or not. Now you have to deploy this model and to the real-time scenarios. Now, to ensure that the model is making prediction quickly and reliably, new patient data is taken as an input and see if it is predicting as seen in the testing data set. Suppose microRNA, for example, microRNA 145 is found is a tumor suppressor microRNA and is quite frequently downregulated in cancer cases. So, as in tested set, it was found that the microRNA 145 was highly downregulated in stage 4 cancer. So when we are entering the data of stage 4 cancer patient in the model which we have developed, so it has to predict that this microRNA expression is frequently downregulated. Then there comes the model monitoring. We have to continuously monitor the model's performance over time to ensure it remains accurate as new data becomes available. Now, what are the challenges and limitations which we face during machine learning? There are several challenges. I have mentioned few of them. First one is the accuracy of prediction. It depends heavily on the quality and quantity of the data used for training. Predictions may need experimental validation because these machine learning models, they require large data sets. So these large data sets are publicly available. We are not providing an experimental data set, but few researchers also go through high throughput screening techniques and they generate the data within their lab, then perform the machine learning models. Then there is complexity of biological systems. So one has to consider the mRNA interactions and complex and context dependent meaning predictions. For example, single microRNA is known to regulate thousands of mRNA. So you need to know the relationship between on it, so as you can signify the importance of the microRNA that you are taking into consideration. So these prediction may vary in different cellular condition or tissue types, so it has to be considered. Because these databases are based on single data set, so you need to consider those. Then there is model training data. Insufficient or biased data can affect the accuracy of the predictions. So this is all for today. I hope everyone had got a brief or understanding of this topic. So let's continue to the Q&A session. Over to you, Sakshi ma'am.
Yeah. Thank you so much, ma'am. It was very informative session. So let's take some questions.
Okay.
Can these sequence two don't not identify cancer subtypes? I thought both ETQR and ETSeqR has same utility, only the behinds method is different.
It can identify cancer subtypes, but the main utility of these tools is to identify the differential microRNA expression patterns. It can be your cancer subtypes in different cancer subtypes also, and between cancerous and non-cancerous tissue samples also.
What is the pipeline for obtaining miRNA data from the patient in a clinical setting? So I think this is the most, everyone wants to know how to get that data actually. Yes.
That data in case of as I would like to explain the experimental protocol to it. First you have to, if you are looking into the tissue samples of the patient, then you have to retrieve tissue samples. You can isolate this miRNA from blood and urine samples also, depending upon the type of cancer you are going to study. So once this biofluids or tissue samples are available, you have to isolate the total RNA, then do the cDNA synthesis. Then lastly, you can do the miRNA expression analysis through RT-PCR. So this is how we get the miRNA data in clinical setting. You can also get data sets also online. There is TCGA data set available, and geo data sets are also available online if you don't want to go for the experimental part.
Mhm. Yes. Next question. Good morning, ma'am. Can ML models handle the high dimensionality of miRNA expression datasets?
Yes, they can handle it. You have to pre-process your data before performing them on any machine learning model. So this high dimensionality is taken care of in the data pre-processing part.
Okay. Next question. What makes miRNAs a suitable candidate for early cancer diagnosis compared to the other biomarkers?
Because miRNAs are pleiotropic in nature, because single miRNA is controlling thousand number of genes. So if you are targeting one miRNA, you are going to control that n number of genes ultimately, and these genes are involved in various processes of cancer like proliferation, and they surpass the apoptotic cycle also. So it's important to target the miRNAs.
Correct. Yes. Next question, please. What type of data are required for training machine learning models in miRNA based cancer diagnosis?
Okay, so the data is basically miRNA expression data. So miRNA expression data could be found, as I said, in online available data sets like TCGA and geo data sets. If you want to identify only the type of miRNA that is associated with a specific cancer that you want to study, then you can look into MIR2 disease, where it gives you a list of miRNAs that are upregulated and downregulated in specific cancers. And HMDD is also, recently updated database. It was updated in 2022, so you can use that database also.
Mhm. Okay. Any other questions do we have? No, I think we have done with the questions and answers. So thank you so much, ma'am, once again for answering all these questions and for the great presentation. Now it's time for the certification of participation and the memento for our guest of honor. It was really pleasure to have you with us, ma'am, today. So we are grateful that you could have joined us today. So again, thank you to all my viewers, and you could please be connect with us. Stay tuned, like us, subscribe us, and be ready for the next session. What we are going to have. So let's connect and grow together. Thank you. Thank you all.
Thank,
Thank you so
thank
much.
you.
Thank you.